Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond.
Welcome back to Neuroverse. Today's episode is about deep mutational scanning and how to interrogate protein behavior. And to discuss this fascinating topic, we are joined by Isabelle Zane, who is a PhD student at the Wellcome Sanger Institute in Cambridge. Having completed a bachelor's in biochemistry followed by a master's in bioinformatics at Imperial College London, Isabelle joined the Parts group at Sanger to study how gene mutations affect the binding interfaces of transcription factors. And today we'll be discussing with Isabelle the process of deep mutational scanning, a method used to systematically study the effects of gene mutations on protein structure and function, as well as how proteins fold, how this process is regulated, and more about the complex dynamics of proteins. Thank you so much for joining us today, Isabelle.
Thank you for having me.
So why don't we begin by you explaining a little bit about what deep mutational scanning is and how it works?
So, deep mutational scanning is a sort of newish method of studying how genotype and phenotype of a gene is linked together. So, deep mutational scanning is basically creating mutations in proteins at a very large scale so that, for example, you would create every possible mutation in the protein one at a time to measure things like how the mutations affect the stability and folding and binding and activity, basically anything you want to measure about the protein. So deep mutational scanning is really interesting because it's a very high throughput method. So in the past, if you wanted to study the effect of mutations, you would maybe create a single mutation in a protein or a few, and then only measure the effect of those mutations. But how do you know which mutations are worth testing, or how do you know which ones are actually going to be informative to measure? But deep mutational scanning allows you to do basically all mutations in a very comprehensive way. And the reason we're able to do this now is because we have methods like next generation sequencing, so you can measure the effect of many, many mutations at once, and also things like very cheap DNA synthesis so that you can design your mutants very easily and for cheap. And the way that deep mutational typically works is that you first create a mutant library. So that's the first step. And then the second step is to apply a functional assay. So then you would select for a particular behavior of the protein that you would like to measure, so like folding or stability or enzymatic activity. And then the third step would be to do the next generation sequencing to actually measure the difference in behavior between the protein variants.
That sounds really amazing, but it seems to me like in a genome of a protein, you would be able to do an infinite number of mutations. So can you really do an infinite number of gene mutations, or do you still need to select the ones that are more likely to influence the phenotype?
Yeah, so most of the time you only do these mutations one at a time. So if you had a protein of, let's say, 100 amino acids long, and because there are 20 different amino acids, and one amino acid would be the wild type, then you would have a library of 19 times 100 in size, so 2000, because then you would have like every possible mutation and every position of the protein, but you only create them one at a time rather than creating multiple mutations at the same time, which increases the combinatorial complexity by way more. But another thing to say is that deep mutational scanning is cool because you're investigating the effect of all mutations, all possible single mutations at a time. You can obviously do more, but the most basic version is to do one at a time, is that you can then measure the effect of mutations that we don't normally see in nature. So if you, for example, just looked at the protein sequence for like a particular protein that's involved in disease across the human population, you might find only certain mutations existing because maybe you just haven't got enough patient samples, or maybe like some mutations are just so deleterious that they don't even show up in the human population sample. But with deep mutational scanning in the lab, you can then generate all the mutations for that protein, even if they haven't actually existed in nature before.
So because of the nature of the number of mutations you can do depends on the size of the protein. is it then more common for experiments to be done on smaller proteins?
Yeah, that's a good point. I think at the moment, at least that I know of, we're kind of only doing single proteins, like probably not complexes at the moment, like large protein complexes that have many subunits. And yeah, there's definitely a case where larger proteins can maybe have lower dynamic ranges measured by the experiment just because they're larger, or maybe the mutations don't affect large proteins the same way. So, for example, a smaller protein will have fewer core residues, like fewer hydrophobic residues in the core because it's smaller, but a larger protein will have many more core residues. So the dynamic range for different size proteins can be different.
So it's really cool that you have this very high throughput method. When you go from just doing the initial deep mutational scanning to picking those that you want to do functional assays on and seeing the effect that these mutations have on the proteins, how do you know which ones to pick? Does the deep mutational scanning give you immediately an indication of which mutations are most likely to cause the greatest degree of instability?
Yeah, so maybe we could go through how you would design a typical deep mutational scanning experiment, and then we can like think about what are the considerations in the process of what you're actually measuring. So like I said earlier, the first step is typically to create a mutant library. And this is actually quite a simple step. So you basically just order the oligonucleotide sequences. So you have the wild type sequence of a gene, and you can order that sequence with your designed mutation inside of them from a DNA synthesis company, and they ship it to you. And then by cloning it into a plasmid, then you have a bunch of plasmids, each one with a different mutation. And then the second step, which is the functional assay. So that's where you kind of decide what you would like to measure. So this is kind of like a important experimental design choice. So if you wanted to measure stability, you might design the experiment a little differently than if you wanted to measure binding. But overall, the idea is you kind of want to link the phenotype that you're trying to measure, so stability or binding or whatever, to something that's easily measurable. So, for example, cell growth is very easy. So, for example, you can design on your plasmid like a mini sort of gene circuit that if the protein binds this other molecule well, then the cell will grow faster. And then so you've converted like this protein stability or protein binding problem into a cell growth problem, so that when you then measure the number of variants in those cells, you basically can correlate the number of cells that contain that variant to the binding or stability or activity of that variant. So that makes sense.
Yeah, that's really cool. It definitely shows the importance of having a specific question and how this method really allows you to test that like one specific cell or one specific protein that you're interested in and seeing the effect that it has.
Yeah, it's really cool. So, yeah.
Yeah, I really like how systematic this approach is. But can you clarify a bit, like what type of model system are you using when you look at these cells? Like what type of cells are they? And also when it comes to protein binding, it seems to me like it's quite a reduced system where you just have the gene of interest of this specific protein. So how much can you look at more complex behaviors like protein-protein interactions, things like that?
Yeah, again, it depends on what you would like to measure. But for example, if you wanted to do deep mutational scanning on like a protein that only exists in eukaryotes, then you're kind of limited to eukaryotes as your model system. If you have like a simple protein that bacteria can handle and express as well in bacteria, then bacteria might be a better choice because they grow faster and are easier to handle. But if you had something more complicated, like something that involves complicated splicing behavior or something, then maybe you would choose a mammalian cell system to do deep mutational scanning in. And I guess you're correct that you're basically taking the protein that you're studying and like putting it into a simplified model system. But in some ways that actually helps you to simplify the measurement so that you know what you're measuring is only protein binding, like basically as simple as a system as possible to measure like this behavior that you're interested in, because you could imagine if you did this in a more complicated cell environment, maybe there are a lot of other confounding factors in the cell environment that might be affecting what you measure, but it would be hard to sort of untangle those effects. So in some ways, it is actually nice to have a simple model system to measure these binding behaviors in.
But then do the cells just express this single protein, or do they express more than one protein?
You mean like, do they express like their native proteins, do you
Yeah,
mean? Or
yeah.
So yeah, the cell expresses all of the normal proteins that it has, but because you've introduced like an extra protein in the form of a plasmid, then it also expresses this extra protein that is encoded on the plasmid.
Okay.
Yeah, and then so you have your functional assay, so something that applies basically a selection pressure onto your protein library so that the good variants perform well and the bad variants perform worse. And then in order to actually calculate the difference in fitness between the good and bad variants, you would need to sequence the cell population before and after applying the selection pressure so that you can see whether the variants increase in frequency or decrease in frequency. And then you would be able to infer that variants that have increased in frequency are more fit because you've linked the good fitness proteins to increase cell growth so that proteins with better fitness will allow the cells to grow faster, divide faster, so they'll be present in the cell population more. So, for example, let's say you want to measure protein stability. So, whether the protein folds into like a stable conformation in the cell. And because the cell has mechanisms for degrading unstable proteins, then you can sort of link the stability to the cell's dependence on this protein. So, for example, you have the protein that you're interested in mutating, and you express that as a fusion protein to an essential gene or protein that the cell needs to survive. If your protein of interest is unstable, then the cell will degrade the unstable protein along with the essential protein that's linked to it, and then the cell won't be able to grow and replicate. But if the protein mutant folds stably, then the protein that it's fused to will also remain in the cell because the cell won't degrade it. So then that cell will have the essential enzyme that it needs to keep growing. Another example could be like for a transcription factor, or just basically linking the behavior of binding to the expression of a antibiotic resistance gene or some kind of drug. So then only if binding occurs between these two things will there be expression of like an antibiotic resistance gene or like some kind of enzyme that metabolizes a drug in the cell culture, or it could even be a reporter gene, like expression of a fluorescence protein, but then you would have to sort the cells. So yeah, you can link it to like any sort of reporter gene. So let me give an example from one of the labs that I work in. We use a system that's based on yeast. So this yeast is basically unable to grow in media where there's like this particular drug present. But if you link your protein library to an enzyme that can metabolize that drug, then any protein that exists stably in the cell, and because it's expressed in fusion with that essential enzyme to metabolize the drug, then the cells can only grow if your protein mutant is stable in the cell.
Yeah. Okay, that makes sense. So you're selecting for the cells that have the correct form of the protein.
Yeah. So the selection pressure is like the addition of a drug that the cells can't grow in.
Yeah.
And if the protein mutant is stable, then it will also express the essential enzyme that can metabolize that drug.
Yeah, I find this experiment really cool. And it also raises the question for me, though, what about cells that may not destroy the unfolded protein and that potentially the unfolded protein causes havoc in other ways? Like examples that come to mind are potentially cancerous cells or like amyloid beta or any other unfolded proteins where the cells should destroy them, but due to some potential misregulation it just doesn't.
Yeah, I mean, that's something that you would try to avoid by picking like a very simple cell as your host system. So you wouldn't want to choose a cancer cell unless the behavior you're trying to measure is specifically like relevant to that cancer cell. So you would choose like a cell system that generally does have these um like degradation systems, or yeah, you would choose the cell system that can give you the readout that you're looking for. But yeah, I mean, there are definitely cases where just because the cell is stable doesn't necessarily mean it has a higher fitness than the wild type, because you're kind of measuring these like biophysical behaviors of the cell, and you might miss out on other things like whether this is actually detrimental to disease or like whether it actually has a clinically relevant phenotype. So just because you have information on protein stability and binding, like you're actually still missing a layer of information on whether that's clinically relevant.
Hmm.
Yeah, I think that raises this super interesting point that neuroscientists especially think about all the time about layers of scale, I guess, because it's really cool in these examples that you've just described that you're linking the protein behavior with the cell behavior. But like you said, this might not be relevant in a clinical context because also as someone who studies plasticity, there may be homeostatic mechanisms that come into play to try to compensate and overcome these changes in the proteins. And then these homeostatic mechanisms could actually be the ones that lead to those clinical phenotypes, or the cell-to-cell interactions may also be different in a fully functioning system, and etc. But this is something we always end up saying when we talk about biochemistry, that it really enforces the need for using a simplified or reduced system to just isolate and ask very specific questions about the function of a specific protein or even the function of a specific amino acid within a protein.
Yeah, I definitely agree. Like basically trying to remove as many like confounding factors in the experiment as possible so that you have like a very clear measurement that the thing you're trying to measure is actually what you're trying to measure.
And as well as having functional assays that look at cell growth or number, etc., could you tell us a bit about what sort of assays you could have for looking at protein structure itself?
Yeah, so there are some really cool ways to measure protein structure. So I think a really cool example is X-ray crystallography. And so if you know about the protein data bank, so that's basically a database where people deposit their protein structures once they've been solved. And it's also the database that AlphaFold models have been trained on in order to be able to predict structure from sequence. So it's basically like a very valuable repository of information about protein structure. And most of the structures deposited in the protein data bank were solved using X-ray crystallography. And yeah, it's actually a very biophysical method. So you basically try and crystallize your protein structures. So you basically want to have a lattice, like a three-dimensional grid where each protein is like one point in the lattice. And ideally, you would want those proteins to be arranged in exactly the same orientation in every point in the lattice so that you have like a lattice of proteins. And the reason why X-rays are used is because X-rays have a wavelength that is very similar to the length of the atomic bonds in proteins. So, for example, like the carbon-hydrogen bonds and the carbon-carbon bonds, which is what makes up most of protein structures, have a similar length to a single wavelength of an X-ray. And that's really useful because you need the X-ray to have a similar sort of length to the thing you're trying to measure in order to have good resolution. So if you had a wavelength that was longer than these biologically relevant bonds, you wouldn't actually be able to like measure the biologically relevant information there. But yeah, so you basically have your protein crystal and you shine an X-ray beam onto that protein crystal, and then when that happens, the protein crystal basically diffracts the X-ray beam and it creates this diffraction pattern. So if you've seen the very famous image of the DNA diffraction pattern that's associated with Watson and Crick and Franklin. That's what a typical diffraction pattern looks like. It's basically a bunch of spots. So that diffraction pattern basically contains information on how the protein has scattered the X-rays onto this detector that creates the diffraction pattern. And depending on where those diffraction patterns are, you can sort of calculate backwards to the electron density of a protein. So you would want to collect as many diffraction patterns as possible so that you have as much information as possible to calculate the electron density of the protein from like multiple different angles. And yeah, and you can't really do this with a single protein because the signal from a single protein, the diffraction pattern wouldn't be strong enough. But by having it in a crystal lattice, you're basically amplifying the diffraction signal by however many proteins there are in the crystal lattice. And so you get a really strong diffraction pattern, and that's why it's useful to have a protein crystal.
That's so cool.
Yeah.
I have a quick question about the diffraction pattern and like basically how do you work backwards into finding the crystal structure? Do you use like a Fourier inversion of the diffraction or how do you go about it?
Yeah, so exactly what you said. There's like a lot of complicated equations, but yeah, essentially it is a Fourier transform. And yeah, because you can work out certain things about the protein crystal based on the diffraction pattern. So the person who figured this out is this guy called Sir Lawrence Bragg, and he used to work in Cambridge as well. And he basically figured out that when you have two waves that are in phase with each other, those waves can be added up to create a stronger signal, right? So the amplitudes of those two waves can be added together. Basically, the amplitude will be double if those waves are exactly in sync with each other, like in the same sort of phase. So that's a constructive wave. So you can also have destructive waves where if the waves are perfectly out of sync, so where the peak of one wave is exactly where the trough of the other wave is, and when you add those amplitudes together, you basically get zero. So because those waves cancel out. And so Sir Lawrence Bragg basically worked out the mathematical details where you will only see the diffraction pattern, or you will only see those waves when those waves are exactly in phase with each other. And so if you can see the diffraction pattern, it means all the waves that have diffracted are constructive with each other. So they're all adding up to something, which means the proteins are like a certain amount of distance away from each other that add up to like a whole number of wavelengths from each other. If it wasn't a whole number of wavelengths, then the diffraction waves would be out of sync and the waves would basically cancel out. So if you just imagine two proteins that are next to each other and you imagine shining an X-ray on those two proteins, you're not shining it directly straight onto them. You're shining it onto them from an angle. The amount of length the X-ray has to travel in order to reach the first protein will be different to the length that has to travel to reach the second protein. But when those proteins refract the X-ray beam, it will change the angle that these beams travel and also change the distance that they have to travel in order to reach the detector. And so those waves can either be in phase or out of phase with each other. So basically, there are only certain angles where shining the X-ray onto these two proteins will actually give a diffraction pattern. Okay, so to link it back to deep mutational scanning, so one of the annoying things about X-ray crystals is that you need quite a large amount of proteins in order to make these crystals. So you need like milligram amounts of protein. And you would need to like find a way to express that amount of protein and purify it. And also it's really difficult to crystallize certain proteins. And you actually don't know before you crystallize it, like what are the best conditions to allow your particular protein to crystallize, because every protein might have slightly different preferred conditions for crystallizing. So even though X-ray crystallography has given us like so many protein structures, yeah, there are limitations. So one of the cool things about deep mutational scanning is that you can sort of interrogate structural things about the protein without needing to crystallize it. And of course, it doesn't fully substitute the need for crystal structures. But for example, with deep mutational scanning, so what the data looks like at the end is you essentially end up with like a heat map where on the x-axis is the length of your protein, so every position in your protein from 1 to 100, let's say. And then on the y-axis is every amino acid. And so then you have this matrix where you can see for every position of that protein what a substitution to a particular amino acid would look like. And you kind of like encode it like a heat map. Can you visualize that?
No, because proteins have 3D structures.
Yes, but this heat map is just on the 2D structure, like just on the sequence itself.
Ah, okay. Yeah. Okay, yeah, that makes sense.
Yeah. So then you have this heat map, which shows for every position in the protein what a mutation to a different amino acid would look like how that changes the fitness of the protein. So that's kind of the data that you get, but you can also map this fitness information onto the 3D structure of the protein. So let's say if you take the average fitness of all mutations for a particular position, so then you get one value for every position in the protein, and then you map that value onto the three-dimensional structure, you can actually identify like surfaces on the protein that maybe all are very important to the protein's function because any mutation on that surface would result in worse fitness for that protein. And so this is really useful because you can identify important surfaces that you didn't know before, and you also wouldn't be able to tell from a crystal structure. And then you could maybe design a drug to target that new surface. And so that's how people sort of use deep mutational scanning to, yeah, design new drugs for protein targets that are previously thought to be undruggable.
But so as far as I understand, all the mutations you create are of a single amino acid.
Yes.
Can a change in a single amino acid actually change at the larger scale the structure of the whole protein?
Yeah, that's a great question. So, yeah, you're right that the most simple experiments only involve a single mutation at a time. And you're also right in that a single mutation might not cause a large enough effect on the protein. But you can imagine like there are certain mutations that are very detrimental to the protein that you can measure with just a single mutation. So a classic example would be the core of a protein structure. So you think of the 3D structure of a protein. Most commonly, the hydrophilic residues would all be on the surface of the protein because the cell environment is aqueous and there's a lot of water molecules, so it's energetically favorable for the protein to have all the hydrophobic residues in the core of the protein and all of the hydrophilic residues on the surface of the protein. But if you make a mutation, for example, that converts a hydrophilic residue on the surface of the protein into a hydrophobic one, it's very energetically unfavorable, and the protein will want to fold in a way that buries that residue in the core, even though in the wild type it's supposed to be on the surface. And that would be like very detrimental to protein fitness. And that is an example of how a single mutation can affect the behavior that you measure.
Mm-hmm.
But also, you can do experiments where you do multiple mutations at a time. It's just that the complexity scales very, very quickly. So I think for those kinds of experiments, you would still have to be slightly selective on which positions you're willing to do combinatorial mutations on. But this brings up a really interesting way to analyze these kinds of data, which is epistasis of mutational effects, like whether two mutations create a bigger effect than the sum of those two mutations when they happen independently. Maybe one mutation is detrimental, but if you make a second mutation at the same time that compensates for that detrimental effect, then maybe the effects cancel out. And so two mutations is actually fit and normal, but one mutation by itself isn't. Or you can also have in the other direction. So you could have one mutation that's good for the protein, another mutation that is also good for the protein, but like when they happen together, they are somehow worse for the protein, or like you just don't know exactly how different mutations at the same time can affect protein behavior. And I think that's really cool because sequences can adapt in very like unpredictable ways.
Yeah, that's so interesting. And do you know how often mutations in nature occur as a single mutation compared to multiple mutations?
Mm. This is really cool because this is sort of the basis of models like AlphaFold that try and predict protein structure. So they use this idea of coevolution. So the theory is that mutations that sort of occur at the same rate must be close together in the protein structure because they kind of have to like compensate for each other. And mutations that sort of are far apart in the structure will have different mutation rates because they're not physically close together in 3D space. So their mutation rates will not have any sort of relationship. And so by looking at the coevolution of residues, that's how you sort of infer which residues are close together in 3D space and how you predict protein structure from protein sequence. But also it's interesting because in nature, you can see proteins that have only 20% sequence identity, meaning only 20% of the residues in those protein sequences are identical, but they might have the same overall fold. So it's like you can have very little sequence identity from each other, but still have the same fold. And that's why it's actually quite difficult to predict beforehand whether a mutation will impact the fold or not, because yeah, sequences just behave in unpredictable ways.
But if you have the same sequence, can you only have one fold?
I think typically you would only have one fold because protein folding theory is that the protein's native conformation is the most thermodynamically stable fold for that protein to have. So the reason why we think you can predict protein structure from sequence is because there's like a single defined 3D conformation for a protein structure. And because there's an old experiment that tested this, which is basically the basis of protein folding theory. It's called the Anfinsen experiment, I think. And basically, the scientist took a protein and then added a bunch of denaturant chemicals to it. So the protein started off in its like very defined conformational state. He added a bunch of denaturing proteins to it, which basically means breaking like the chemical interactions that the protein has folded into. So basically turning the three-dimensional structure into like an uncoiled sort of like spaghetti mess of the protein chain. But then when you slowly remove the denaturant chemicals, you actually recover the original activity of the enzyme that you denatured, meaning that the protein has like recovered its fold. And all he did was remove the denaturants from the solution. And so, yeah, without any sort of knowledge of what the protein structure is supposed to be like, the protein somehow ended up at the same structure that allowed it to carry out its enzymatic activity. So, yeah, in a sense, each sequence defines like a uniquely energetically favorable conformation for that protein.
Very cool.
Is it okay if we go back to the other techniques that you use to understand the protein structure, such as NMR and cryo-electron microscopy? So you've spoken about like the challenges of X-ray crystallography and how we don't know in advance what's the optimal way to crystallize them, and how the deep mutational scanning helps inform this as well. So do you use NMR or cryo-EM, and how are these compatible with the deep mutational scanning?
So yeah, those are also really cool methods. So NMR, which is nuclear magnetic resonance. So another limitation of X-ray structures is the crystals are actually frozen. So like the proteins are stuck in one conformational state, like one structure. But in reality, proteins in the cell are dynamic and like moving all the time, like most proteins are. So most proteins are actually like their behavior sort of depends on being able to move, having loops or like globs of the protein that fold over each other and like carry out their activity. And so you can't really see that on a protein crystal because the crystal has to have like its defined shape and it's frozen. But something like NMR, the proteins are measured in solution. And NMR is it's all about the spin of the particles in the protein. So all fundamental particles have like an inherent spin. So, and spin of charged particles, like electrons, create a magnetic field. So it's kind of like the Earth, where you have like the spin that creates in the magnetic north and south poles. And so the protein has this spin, like all of the atoms in the protein has its spin, but you basically apply a separate magnetic field to the protein, which some of the particles in the protein will absorb a radio frequency pulse that you apply to it, and that changes the spin of the atoms in the protein. And you measure how much of this was absorbed. And so that kind of tells you, because the spin of the atoms in the protein are also affected by which atoms it's around. It's all sort of magnetic. So atoms that are close together in the protein will have coupled spins because they're close together. And so by very complicated, also Fourier transform stuff, you can essentially work out which atoms are bonded to each other and which atoms are close together in three-dimensional space. But basically, because the proteins are in solution, and because proteins are dynamic inherently, so what you end up with is a series of structures. So you call it an ensemble of structures. Each one is slightly different to each other, but you kind of like average out that ensemble of structures to get one eventual structure. And so NMR structures, if you look at them on the PDB, they basically look like 100 superimposed structures, all like superimposed onto the same 3D space, and they look like a crazy noodle-like image. But yeah, so that's really cool as well, and that does allow you to look at protein dynamics more. And for cryo-EM, that's a newer technique compared to NMR and X-ray crystallography, and it's getting very popular because essentially you don't have to make crystal structures that are as difficult as you do in X-ray crystallography. You can often do like single molecule things with cryo-EM, and so that allows you to, avoid the difficult crystallization. But the physics of it are basically you have an electron beam that points at the protein that you're trying to image, and you end up with an electron sort of micrograph, and you can analyze that in many different ways. But it's really good for these like large protein structures that would be really difficult to crystallize with X-ray crystallography. And there are other methods too where you can look at protein dynamics. So there's something called serial femtosecond crystallography, and that allows you to look at proteins on the femtosecond scale. So femtosecond as in like a very, very, very short time scale that is about the same time scale that protein dynamics happen. And so with this method, you can actually see the protein moving, but it's a very, very new method, and it's not really widely used yet. And yeah, so I guess deep mutational scanning you can't see this like level of exactly what the protein is doing. You're kind of inferring based on the fitness measures whether the protein is performing well or not. But yeah, you're not actually ever seeing the structure or the dynamics of the protein. These are all inferred from your experimental data.
Yeah, thanks for explaining the methods in detail. When you were explaining NMR, I kept thinking like, wow, this is so similar to MRI, which is magnetic resonance imaging, which they usually use to image tissue or like the brain or various parts of the body. And then I realized like it literally is the same thing because both techniques are based on magnetic resonance and the spin of electrons.
Yeah, I think when I had lectures on NMR in undergrad, they said that they changed it to be called MRI in the clinical setting because patients would be scared of having the word nuclear in there, but it's essentially the same thing.
Right. That's cool.
Just out of curiosity, with the deep mutational scanning, do you think that is there some sort of, I don't know, tool or platform where similar to the protein data bank where you can like basically download the blast sequence and then have the XY coordinates of the proteins versus the amino acids that it has and do your own heat map and just like play around with it, or do you need to do the functional assays?
Yeah, so there's no sort of like centralized place where people deposit their deep mutational scanning data yet, or that I know of. I know there are researchers who are trying to make one happen, but it's slightly annoying because every experiment kind of can be done in a different way. Methods are not as standardized as something like X-ray crystallography, where basically every measurement that comes out of an X-ray crystallography experiment is the same. So like they all measure the same sort of parameters for the protein crystal. But with deep mutational scanning, you could have the same proteins measured either for stability or binding, but maybe like the specific way you define stability in one experiment is different from the way it's defined in another experiment because you've applied different selective pressures. And so it makes it really difficult to standardize because they're measured in different experimental conditions. So at the moment, it's not really standardized. So you kind of have to keep that in mind when you're collating deep mutational scanning data sets, which people still do, but they just have to keep in mind that, yeah, it's not necessarily directly comparable.
Yeah, cool. So just going back again to protein structure and stability, could you tell us a little bit about, so after you've done all of these assays and you can look at the cell growth as an indication of how stable the protein is, can you tell us a little bit about how the protein actually folds inside the cell?
Yeah, so the assay depends on having like cell mechanisms that detect whether proteins are stable in the cell. So like if the protein misfolds or is unstable, like the experiments kind of rely on the cell to detect that for us because basically we know that cells degrade unstable proteins or proteins that don't fold properly in the cell. So it's really cool. So the cell has like different molecular chaperones that regulate the process of protein folding.
I'm a big fan of chaperones. One of
Okay.
our early episodes, we did it on structure-function relationships, and I went on and on about chaperones. They're so cool.
Yeah, it's really cool. So one of the chaperones for folding is called the GRO-ES complex. And this is a giant protein complex that bacteria use in order to make sure proteins fold properly in the cell. And eukaryotic cells have their own versions of this complex. And basically, the GRO-ES complex, so the way you spell that is GRO and then capital EL or GRO capital ES. It basically looks like this giant barrel with like a lid. So it's a really large protein. So it's like almost 200 angstroms in length, but the average length of a protein is about like 60 angstroms. So basically, it's just a giant barrel, and its role is to sort of, um, it basically it's the interior of the barrel has hydrophobic patches. And as I mentioned earlier, when a cell is properly folded, most of the hydrophobic residues will be inside the core of the protein because it's energetically unfavorable for hydrophobic residues to be interacting with water. And so these hydrophobic patches inside the GRO-EL barrel basically binds to proteins that don't fold properly and have their hydrophobic patches exposed. And then the lid of this barrel, which is the GRO-ES part of this complex, it basically traps the unfolded protein into the barrel, but the binding of the lid to the barrel actually changes the shape of the barrel. So proteins function through these really cool conformation changes, and this is one of them. So binding of the GRO-ES lid to the GRO-EL barrel essentially causes a change in the GRO-EL barrel that makes the hydrophobic patches in the barrel disappear and get replaced with hydrophilic surfaces. So now the unfolded protein, so this is a single unfolded protein that this would happen to. The single unfolded protein is trapped inside this barrel and doesn't have any like hydrophobic surfaces in the barrel to stick to because the barrel has changed to remove those patches. And so that protein is basically allowed to fold into its native state because the barrel basically separates it from the rest of the cell environment. So it prevents anything else in the cell from interfering with the folding process. And so the unfolded protein basically is forced to refold until it finds its naturally energetically favorable state. And then when that happens, the lid is released along with the properly folded protein. And then because the lid is released, the conformation change of the lid binding is reversed. And then you have hydrophobic patches inside the barrel exposed again, and then a new unfolded protein can come and bind. So like it's just fascinating how something like this has evolved in the cell. Like it's a very highly regulated process. Yeah, it's just an amazing complex, really.
Yeah, but also so like self-regulated, like a machinery that just does its thing again and again. Yeah, that's so cool. On that note, we'll end today's episode, which was a really fascinating delve into the details of how proteins fold. Thank you so much, Isabelle, for joining us today and sharing all of your knowledge.
Thank you so much for having me.
Yeah, it's been super fun.
Isabelle also draws a series of cartoons that's called Cellmates, which is inspired by people and things in the lab. And so I highly recommend checking it out. You can find it on her Twitter, which is at Isabellease, Isabellease. And thank you for listening to the episode. We hope you enjoyed it, and please feel free to check us out on social media at neuroverse_pod on Instagram and Twitter, and also check out our website, neuroversepod.com, to find out more.